Okay, we're going to break C-Lab tradition and actually get started on time today so that we have time for questions. And Jim has a pretty busy schedule today. Before we get started, though, I wanted to just acknowledge there's a lot of effort that goes into hosting these. I want to acknowledge Dr. Jeff Krause, the co-chair of this committee with me. And I'd like to thank our sponsors, the Dauphin Island C-Lab, the University of South Alabama Department of Marine Sciences, and then the C-Lab Foundation and the USA Foundation. And finally, our benefactors for this, the late Pete and Marty Wiesa. I think they would be very pleased with the speaker that we have today and very interested in Jim's work. I'd also like to thank Julie Herman from the Department of Marine Sciences and Angela Levins from the C-Lab for all of their work, and then finally the graduate student organization for all the help that they've given us to coordinate the logistics. So for introductions, it's not often that you get to introduce one of your science heroes, but I've been following Jim's work since I was a graduate student. Jim is one of the preeminent estuarine scientists in our field. He's been very productive over the years. He's been at the USGS for 4 decades, and he's racked up some pretty remarkable accomplishments over that time. His work, foremost, he's been cited over 17,000 times, which is pretty incredible. He's had a long list of honors and awards. Some of the most recent ones include the Odom Lifetime Achievement Award from the Coastal and Estuarine Research Foundation. He's been named a fellow for the American Geophysical Union, as well as for the Association for the Sciences of Limnology and Oceanography. And he's also won the Ruth Patrick Award, a Lifetime Achievement Award given by the Association of the Sciences for Limnology and Oceanography. He's been a really great mentor over the year. He's hosted over 13 postdocs. Actually, one of his former postdocs, Dr. Jane Caffrey, is here today. She came over from Florida. And he's been served on over 27 graduate student committees. In addition to his science recognition, he's also been recognized for his service. And I think what really sets Jim apart from a lot of scientists is that he's really been a great translator of science for policy and management, and he's been recognized with the Distinguished Service Award from the Department of Interior twice for this work. As a former federal scientist myself, I can attest to how difficult it can be to remain scientifically relevant, as well as to do good work in translating and working with policy and managers. And I think Jim has really been one of the best in terms of doing this. Let's see. I could go on and on here. Personally, I think Jim, really one of his seminal works was a paper that he published in 2001 titled "Our Evolving Conceptual Model of the Coastal Eutrophication Problem." This paper alone has been cited over 2,600 times, and I still pull it out every once in a while just to get myself back on track and to get some inspiration. It's really a terrific piece of work. Let's see. I think another thing that's been terrific about his career is his focus in using San Francisco Bay as a research laboratory. By working on that system for over 4 decades, he's really been able to unlock the understanding of that system, really understanding the ecosystem and global change, watershed change, as he talked about in his seminar last night. But I think what's been really remarkable is how he's been able to generalize those patterns and processes worldwide. So not only just applying them to San Francisco Bay, but through modeling and literature, putting together data synthesis, he's been able to apply those. And one of his recent papers I'm really a big fan of that was published in Water Research, where he looked at why large phytoplankton dominate in estuaries. And I told him last night, it's like one of those where it seems like a really simple idea, and it's kicked myself. It's like, why didn't I think of putting this together? But it's very elegant, and he was able to generalize from San Francisco Bay to kind of global patterns of what we will expect in the future. So we're extremely grateful to have Jim here, and please join me in welcoming Jim as our 21st WESL lecturer.
Thank you, John. Can you hear me okay in the back? I felt a really warm welcome at this institution, and I really appreciate the hospitality. When I started studying and observing San Francisco Bay in 1976, we knew very little about that estuary. In fact, the word estuary wasn't in the 1970s. And so we started in salinity and temperature and phytoplankton biomass in communities, and we're interested in things like seasonal patterns. And after we had collected data and made observations for a number of years, we started thinking interest between wet, high flow and dry, low flow years. And this continued for a while, and at one point I looked back and I thought, you know, we've made 20 years of measurements in San Francisco Bay, and we've seen some surprising changes. And now, from the beginning of that program that started before I arrived, we've collected 50 years of measurements in San Francisco Bay. And this estuary is very different from the estuary that existed when I started studying it in 1976. And now we can make some statements about patterns and processes of change at timescales longer than a few years or a few cycles of the ENSO system. And since these recent decades have passed, we've accumulated time series of observations in many different estuarine and coastal systems. So we can, our understanding of coastal systems is deep and great. Making measurements over a long processes that fluctuate over periods longer than several years. So that's what this talk is about. This talk is about variability of estuarine coastal ecosystems at time scales longer than several years, and I'm talking about multi-decadal patterns of variability. So the talk is about both patterns of variability, patterns of change, and their underlying processes. So the first part of this talk is I'm just going to show you some patterns of change. I'm going to show you some techniques that we can use to extract information from time series data to characterize patterns. And then in the second part of the talk, I'm going to talk about what steps can we take to analyze environmental time series data to get clues about the processes that are driving change. So the first thing that I want to do is just show you a whole bunch of time series that have been collected of many different kinds of constituents across the world. And I'm not going to say anything about these time series. I just want to show them to you to give you a sense of what I'm talking about from a global perspective. So, bam. Some of the patterns that we see of change over multiple decades indicate patterns of increase over time. So, for example, and I showed this last night, increasing species diversity of fish in the Thames estuary after oxygen returned. Increase in the areal extent of hypoxia in the Baltic Sea. This is one that really intrigues me. Increasing relative proportion of dinoflagellates to the phytoplankton community in the Gulf of Finland. A couple of more upward trends. Warming in the coastal waters of Germany, measured off the island of Helgoland. Increasing nitrate concentrations in the South China Sea. This really remarkable record of verities of bacterial increase in the Skidaway River estuary. So these are examples from around the world of patterns of change that look like trends of increase. And of course, there are an equal number of patterns of change that look like decrease. So this is a pattern of decreasing sediment input to the Yangtze estuary, and this is a result of construction of the Three Gorges Dam. Decreasing nitrogen and phosphorus concentrations in Danish coastal waters, decreasing ammonium in German coastal waters, decreasing pH in Chesapeake Bay that is clearly acidifying. And some of the trends contained in the long time series, well, here's some more downtrends. Decreasing primary production in Tampa Bay after nutrient loads were reduced. A decrease in an index of light scattering, an optical property of Chesapeake Bay, And if phenological change, earlier onset of the cyanobacterial blooms in the Baltic Sea that are developing earlier now because of earlier warming and earlier thermal stratification. And not all of these patterns look linear. Some of these patterns look nonlinear, like this pattern of decreasing mercury concentration in mussels in the 4th estuary, decreasing chlorophyll in Danish coastal waters. And I showed this last night, this almost exponential increase in the number of non-native plant and animal species in San Francisco Bay. So patterns of change don't have to be linear. They don't even have to be monotonic trends of patterns of change over time. Some of these have inflection points. So as an example, we see this decrease of dissolved oxygen in Deep Bay, part of the Hong Kong system, and then an increase. We see this fast ramp up of total phosphorus loading to the Potomac estuary, this steep decline, and parallel with that, decreases in dissolved oxygen and then recovery of dissolved oxygen. I showed this last night. And then I showed part of this graph last night. I showed this part of decreasing shrimp catch in the Mediterranean coastal waters of Egypt. I didn't show the second part because I didn't have time to talk about, but you see this recovery of shrimp catch in Egyptian coastal waters. So there are patterns of change that are not even monotonic, and some of the changes that we capture in observational programs are not steady changes, but rather abrupt, discrete changes over time. So here we see a discrete shift up in the abundance of copepods in the Gulf of Trieste in Italy, this obvious discrete downshift in the number of macrofaunal species in the Bay of Brest, and from our data, this discrete shift up of summer chlorophyll that took place in the southern part of San Francisco Bay. Oh, I know what I did last night. I hit the blank screen. That's what I did. The wrong button. Okay, so the point that I wanted to make with this very quick survey is that change is common in estuarine coastal systems. Virtually everything that we measure, from bacterial abundance to dissolved oxygen to nitrate to sediment supply, virtually everything that we measure is changing. These changes take on many different patterns, and that implies many different processes of change. So with that as kind of a general introduction to the theme of changes in their patterns from a global perspective, now I'd like to dig deeper into changes that we've captured in our observational program. Actually, I'm not going to show any of our data. I'm going to show results that have been captured in another observational program in the estuary of San Francisco Bay. So I showed this last night. This is the regional setting. This large interior part of the state bounded by the Sierra Nevada and Coastal Range Mountains is the watershed. Runoff from that watershed is collected into the San Joaquin River that flows to the north, the Sacramento River that flows from the south. Those rivers converge, and that's the source of fresh water that makes San Francisco Bay an estuary, and the source of seawater is the coastal Pacific Ocean. Now, the state of California has a remarkable monitoring program that goes back to the early 1970s. And for the rest of this talk, I'm going to show you data that have been collected by that program that's run by the California Department of Water Resources at this particular location. So to get you oriented, this is the Sacramento River system. This is the San Joaquin River system. They converge in this region and carry fresh water out towards the coastal ocean. This monitoring program samples at least once a month. I'm going to show you results of surface measurements. They make these measurements across a network of sites in this inland part of the Delta estuarine system. I'm only going to show data from this one location, and I picked this location because it's representative of an estuarine site that oscillates between being fresh, it's a freshwater, tidal freshwater system during high river flow periods, and brackish water during the low flow summer season. First of all, what do the data look like? And I'm going to be talking about time series of 10 different water quality constituents over the period 1978 through 2016. And so we have monthly time series of surface salinity, temperature, suspended particulate matter, this is in milligrams per liter, Secchi depth, chlorophyll A in micrograms per liter, dissolved oxygen as percent saturation, and then 4 different nutrient constituents, the sum of nitrate and nitrate, ammonium, phosphate, and silicate. And when you take a first glance at these time series, this can be kind of overwhelming because there's complexity here. I mean, there's complexity in each of these individual records, and then if we just look at the differences between these time series, there's complexity at that level too. So it can be a bit daunting to try to ask yourself, how can I extract information from these kinds of time series? And one of the key points that I want to make is that these time series are rich in information. There's large information content in these kinds of time series. It's not necessarily obvious how we extract that information. And so what I'd like to do is show you 5 simple steps that you can take to incrementally extract different levels of information from these kinds of time series. So in a sense, this talk is kind of a tutorial. What do we do with time series data? So the first thing that we need to do is look for patterns of variability, quantitative patterns of variability. And I think the single most powerful and useful tool that we have for extracting patterns is our eyes. And so I already said that. So the very first and most useful step is just to visualize the data. And if we're interested in longer-term patterns of variability, a useful step is to smooth out the high-frequency monthly scale variability with some kind of smoother, like a low S function, to help us visualize the patterns of change that have taken place over periods of decades. And I'm showing 3 examples here for ammonium, for chlorophyll A, and for phosphate. So my eyeball test tells me that it looks like there might be a long-term trend of increasing ammonium, but we're not sure. The eyeball test doesn't give us any validation of that inference. It looks pretty clear that there has been a long-term decrease of chlorophyll A, and it looks, the eyeball test tells us that that's not a steady change. It appears to be an abrupt kind of change. And the phosphate series, it's not clear. The eyeball test doesn't tell me anything clear about whether there's a long-term pattern of phosphate variability change over time. But it does look like there are things like inflection points. It looks like the 80s were a period of increasing phosphate, the 90s were a period of decreasing phosphate. So maybe something's going on at time scales longer than the full period of record. So the first step is to just look at the data, use the eyeball test because it's really powerful. The second step that we often do, and oftentimes people stop at this step, and one of the things I want to teach you is if you're working on these kinds of data, don't stop at detecting trends. But there are a number of statistical tests that we can use to determine whether there are trends of change over time, whether they are statistically significant, and what their rates are. Are they positive or negative trends, and what are their rates? And for a variety of reasons, the appropriate tests that we should be using for trends in environmental time series are not parametric tests like linear regression, which is a bad idea for multiple reasons. But instead, there are non-parametric tests that are more appropriate for detecting trends in environmental time series. One of my colleagues at the USGS, Bob Hirsch, is a statistical hydrologist. He uses these non-parametric tests, and his career has been built around detecting changes in variability in hydrologic components of change over time. So there are a number of advantages. These non-parametric tests have greater power of detecting trends in data that are non-normal. Most of the data that we work with are not normally distributed. The seasonal Kendall test, which Bob Hirsch developed, removes the effects of seasonal variability. So any series that has a seasonal component of variability creates problems in the significance level that we assign to trends. And that resolves that problem. And then the slope that we end up with, we want to know the rate of change in addition to whether change is significant or not. The slope is resistant to the effects of extreme values. If you're interested in this topic, I recommend that you read these 2 papers because Bob gives a very clear, detailed explanation of the advantages of non-parametric tests of trend versus parametric tests. So I use the seasonal Kendall test as a test for detecting trends in those 10 environmental time series that I showed you. And I'm going to go back to these 3 time series that I showed you earlier where I started with the eyeball test. So With respect to the ammonium trend, the seasonal Kendall test tells us that there has been a significant increase of ammonium concentration over time. So it validates the conclusions that we drew from the eyeball test. The chlorophyll record also has a significant trend of decrease over this 38-year period. There's no long-term trend in the phosphate concentrations. Although this oscillation here really nagged me, and I didn't want to leave this alone. Okay, so the third step that we can do, and kind of the concept that I want to give you here, is that detecting trends is a starting place, and it gives us one layer of information. But we can uncover other layers of information by doing additional tests. And so I'd like to show you 3 additional tests that we can go that's going to give us more information about these patterns of change over time. And the first is to ask the question, if we have a time series like chlorophyll that has a significant trend of change over time, what was the shape of that pattern of change over time? Was it steady, or did it develop as a discrete change in time? The eyeball test tells us that there was a discrete change in chlorophyll concentrations, and there is a non-parametric, and like trend tests, there are many change point detection tests that we can use to look for periods of a record where the statistical properties of a time series have changed. So for example, either the mean or the variance on one side of the change point is significantly different from the mean or variance of the series after that change point. So these tests look for periods in a record when there are significant differences in statistical properties, and it identifies the point in time when that difference is the largest. So the non-parametric test for change points is called the Pettit test, and I applied that test to the chlorophyll time series, and it identified a change point in the chlorophyll record in the year 1987. So now we know from the seasonal Kendall test that there is a significant decrease of chlorophyll over time. Now from the Pettit test that we know that change happened as a discrete change at one particular point in time. And as we're revealing these different layers of information, we're getting stronger clues about what the underlying causes of these patterns of change are. So patterns can be very helpful in terms of identifying underlying processes. Now, in this case, this is a pretty well-studied estuary. We've known for a couple of decades now what the underlying process of this discrete change was, and it was the introduction of a non-native species. In this case, it was a small clam indigenous to the Western Pacific that was introduced into San Francisco Bay in late 1986. This is either Corbula or Potamacorbula amurensis, and it was first detected in autumn of 1986. Within a year, its population exploded. It carpeted the bottom sediments at this location in the estuary, this low salinity region. This is a filter feeder, and its filtration rate is fast enough to remove phytoplankton biomass from the overlying water column. So once that organism, that filter feeder, became established, dominated the benthic invertebrate community, we had this discrete downshift of chlorophyll concentrations, and largely as a result of the loss of the summer bloom. This is a part of the estuary that had this recurrent large summer phytoplankton bloom. That summer bloom completely disappeared as a result of filtration by this non-native species. So changes can happen as discrete events, and this is a pretty clear example. Okay, so these oscillations of the phosphate record really bugged me, and I wanted to know what's going on here. And so one of the things that we could do, in addition to doing trend tests over the full period of record, we can do a sliding analysis of trend tests over subsets of the record. So I just picked a 15-year window and did incrementally seasonal Kendall tests over subsets of the record. And what that analysis showed was that, yes, in this period of the 80s, there was a significant increase of phosphate concentrations. And yes, after that, there was a period of significant decrease of phosphate concentrations. So these series can have these oscillations, they can have these inflection points. And in terms of processes, probably what was happening here was that this is a period of population growth. The important source of phosphate is an upstream wastewater sewage treatment plant. So as phosphorus loadings from that wastewater treatment plant increased, that explained this period of increasing phosphate downstream in the estuary. And this interesting period of decrease coincided with the period in time when we started banning phosphate-based detergents. And in 1996, California banned phosphate detergents, and so that accounts for this step decrease. So one of the tests that we can do is to do the seasonal Kendall test on subsets to look for oscillations and determine whether they're significant or not, and if they are, try to figure out what the underlying causes of those oscillations are. Okay, now the last additional test that we can do in addition to trend tests over the full period of record. So everything that I've shown you to this point addresses the question, is there a change in the annual mean quantities? Is the annual mean changing over time? What we can also do is trend tests on the series binned into individual months. And so here, the appropriate test is the non-parametric Mann-Kendall test. So, for example, for the chlorophyll series, I did the Mann-Kendall test on the complete series of January mean measurements from 1978 to 2016, and then for February and March and so on. And this graph shows the results of this analysis. Each bar is a slope, and you see that they're all negative, so this says that for every month of the year, chlorophyll has decreased, but the blue bars are statistically significant. So there's no change. There has been no change of chlorophyll during the first part of the year, but really rapid changes during the warm season. And again, this is a result of the disappearance of the summer blooms as a consequence of the introduction of this species. This is the record for salinity. So this is these are the monthly trend tests for salinity. And the feature of this record that stands out to me is positive trends of salinity for the months September, October, November, and December. So appears to be a pattern of increasing salinity, but only in the autumn season, not in other months of the year. And then I did this for all of the 10 constituents, and I'm just going to show two more. This one for ammonium. And so what you see here is another very different, intriguing pattern. So there is an overall trend of increasing ammonium. I showed you that. But most of the action is during the cold seasons, January, February, and December. That's when the increasing rates of ammonium are strongest. And if we look at the summer months, for July and August, there's no trend of ammonium. So this is pretty intriguing. So now we know that there's a trend of ammonium, but it's focused on particular seasons of the year. It's not happening during the summer. It's happening during the cold season. And then for silicate, every month of the year had positive trends of silicate. So silicate concentrations have had an overall increase. We've known this for a while, and we've understood this as loss of the summer blooms. So during the early era, when the summer blooms were recurrent, those blooms were dominated by diatoms, and so the diatoms drew down silicate during the summertime. That process has largely ended. So the assimilation of silicate during the summer months has largely ended, and as a result of that, silicate concentrations have increased over time, and the fastest increases during the warm season when those summer blooms disappeared. So I guess the point again that I want to make here is that there are a number of steps that we can do going beyond simple trend analysis to give us a deeper understanding of how things are changing over time. So that's the part of the talk about patterns. I wanted to give you examples of tests that we could do to extract patterns from monthly time series. I think that one of the most important ones is to break the trends down by month, because there's a lot of information revealed by looking at trends by month. So in terms of processes, now the challenge is, okay, we've got a pretty clear understanding of how things have changed over time. What drove those changes? And well, actually, let me explain one more thing. When we start, I'm going to be talking about a particular kind of statistical model, generalized additive models to look for associations between these water quality constituents. But I think that we all recognize that before we start doing any kind of coding or statistical modeling, we need to develop a conceptual model in our heads of how these changes have occurred. And so so this is my conceptual model of this estuary and how these changes, what might be driving these changes. So those of us who are working in estuaries, the first thing that we think about is river flow as a driver of change. And so there are quantities that are measured that we call freshwater inflow from the Sacramento and the San Joaquin rivers. So this is inflow from the watershed to the estuarine system. Some of that inflow is exported to state and federal water projects, and now on average we're exporting about 30% of that inflow. So we need to know if exports are having an effect on water quality in the estuary. The difference between inflow and export is what we call outflow, and this is basically the net river inflow to the estuary after we subtract exports. I've shown you that corbula had an effect at least on chlorophyll, so its presence or absence is something that we should consider. And then in terms of the nutrients, one of the largest sources of nitrogen and phosphorus is a sewage treatment plant for the city of Sacramento. So a point source discharge here that's pretty far from the downstream sampling site in the estuary. So with that conceptual model in mind, we fortunately have measurements of those components of what we think is driving this variability. So we have records at least dating back to the 1980s of nitrogen loading from that wastewater treatment plant. We have daily records of freshwater inflow from the river system. We have daily records of export, and I'm using export as a fraction of the inflow, and you can see that the water development and water management effects on export had a big increase from the 1960s to the 1980s, and now on average we're exporting about 30% of inflow. And then we have records of the population abundance of corbula. So we've got data on monthly scale variability of 10 water quality constituents. We've got daily or monthly scale variability of the processes that we think are driving this variability. Now the question is, how can we find associations between these different quantities that are being measured? And I used the technique of generalized additive models for a couple of reasons. And one is that it's a tool that allows for nonlinear relationships. And we could expect that many of these relationships between drivers of change and responses to change are nonlinear functions. And so first of all, that's the tool that I use, generalized additive models. Secondly, I didn't use these models to try to understand or identify processes of variability of annual mean quantities, but rather I focused on the times of year when the changes were largest. So rather than trying to understand annual variability and annual mean salinity, I focused on the annual variability of salinity only during the autumn months. So I constructed a new time series of mean September through December salinity. So the idea is to try to understand variability only during those seasons when the changes are largest. Let's see, what else? And the third thing, the modeling strategy was not to find the statistical model that explained the largest proportion of the variance in this case, autumn salinity, but rather to find two quantities that explained the largest, those two quantities that explained the largest fraction of variability of that autumn salinity series. So, if you use generalized additive models and consider all possible drivers of change of autumn salinity, the two factors that stood out as explaining the largest component of variability are, number one, freshwater inflow. And so this is what's called a partial residual plot. It's one of the outputs from this analysis. I did this analysis in the RMS package in R. And these partial residual plots show the shape of the functional relationship between, in this case, autumn salinity and autumn inflow. And the second important quantity that stood out was the fraction of that inflow that's exported. So let's kind of walk through these figures. This figure is telling us something that we could have anticipated. If I'd thought about this a little bit before I did the analysis, I think we could have predicted this relationship. It says that those years with very low inflow during the autumn have high salinity. And that's something that we would expect, because as inflow as inflow becomes small, either seasonally or from year to year, salt intrudes further into the estuary, and this sampling site becomes saltier as a result of saltwater intrusion. As inflow to the estuary increases, that salinity gradient is displaced further downstream, so the salinity decreases at that sampling site, and the curve flattens out, and the curve flattens out where the salinity reaches zero, and so any further incremental increases in inflow are not going to have an effect on salinity because you can't get lower than zero. And then so there's a negative relationship between autumn salinity and autumn inflow. And then if we look at the relationship between autumn salinity and the fraction of that inflow that is exported, this is the water management effect, we see a positive relationship. So those years in which we were exporting a very small part of the freshwater inflow to the delta system were years where salinity was low, and as we increase that fraction of exported water, salinity goes up. So this is pretty strong evidence that a human activity exporting fresh water from the estuary is having an effect on salinity during the autumn. Now, not only does this approach give us an idea of what are the important processes of change, what are the shapes of the relationships, but this particular package also gives us an estimator of the relative importance of these two factors. So now we can say something about the effect size of inflow and the effect size of the fraction of inflow that's exported. So the effect size for inflow, it's negative in sign. We have a negative relationship. The effect size is 5, and the units are the units of salinity. And the effect size of exporting water it's positive, and it's 2. So now we know that the effect of inflow, which is largely a natural climatic process, is about 2.5 times the effect size of exporting fresh water. But this is significant. We're having a significant effect on salinity during the autumn. So here we have a process that just represents the dilution of sea salts by freshwater inflow, and here we have a process of water management of exporting fresh water away from the estuarine system. Now I'm going to do the same kind of analysis for 3 other quantities, and this one is chlorophyll. And you remember the monthly analysis by trends showed that the chlorophyll changes were largest in the summer. So again, for every year, I calculated mean summer chlorophyll, and I tried to see what factors explained variability in mean summer chlorophyll. And we already knew that clams were important, and I treated clam abundance, this is the introduced species, as a categorical variable. It was either present or absent. And that turned out to be the single most important predictor of summer chlorophyll variability, whether this introduced clam was present or not. And so that relationship was negative. So these were the 6 years before the clam was introduced, and so these are the individual summer chlorophyll values. And, oh, by the way, these are, I couldn't find and validate a model of untransformed chlorophyll, and so I used this model on log transformed chlorophyll. So the axis, the units here are log of chlorophyll. So these are the years before the introduction, these are the years after the introduction, and you can see that after the introduction there's a lot of variability in summer chlorophyll. And that variability has to do with the second factor, which is outflow, the amount of freshwater outflow that actually was delivered into the estuary. And this is a relationship also that we could have predicted on the basis of the relationship between outflow and flushing. And so during years of very low summer outflow, the residence time of phytoplankton and other constituents in the estuary is long. That gives time for the phytoplankton community to build. So we have this negative relationship. And then as outflow increases during summer months, chlorophyll concentrations go down. And in this case, we can't, because clam abundance was treated as a categorical variable, we can't use this method to compare the effect sizes of these two components of variability. The next one that I want to show is summer silicate. So again, I constructed a time series of annual mean silicate during the summer months, and it turned out that the most important predictor variables for summer silicate was outflow, another hydrologic measurement, and the second most important was chlorophyll A as a proxy for phytoplankton biomass. And again, these are the shapes of these relationships we kind of could have expected, we could have anticipated this, So the outflow effect during years of very low outflow, those are years when salt intrudes further into the estuary. We know that there's a big gradient of silicate between the rivers and the ocean. Silicate concentrations in the rivers are about 300 micromolar. The source is weathering of silica minerals in the watershed, so the rivers are very enriched in silicate. The coastal ocean has silicate concentrations of about 20 micromolar. So as salt intrudes into the estuary, higher proportion of seawater relative to freshwater, and that seawater has lower silicate concentrations in it. So as outflow increases, the proportion of the high silicate freshwater increases, and the silicate concentrations during the summertime increase. And again, this curve flattens out when the flows are approximately fresh, and further incremental increases in outflow don't change the salinity or the silicate concentration at that location in the estuary. And the inverse relationship with chlorophyll reflects the fact that silicate was drawn down a lot during this pre-clam invasion period when these summer blooms develop. So when chlorophyll used to be high during the summertime, silicate was drawn down to low concentrations, and in the years after the invasion, when those summer blooms disappeared, silicate concentrations have increased. So now we know that the trend of increasing summer silicate is associated primarily with this trend of chlorophyll, because there is no trend of outflow over time. And again, we can compare effect sizes. So this positive relationship between outflow and silicate, and that reflects the fact that the river is a source of silicate. So as river flow goes up, the silicate concentrations go up in the estuary. And the effect size is positive, and it's 38. And the effect size with respect to chlorophyll is negative, and it's minus 21. So the hydrologic component of variability, the riverine source, explains about twice as much of the annual variability of summer silicate than the algal uptake term using chlorophyll as a proxy explains. The last example I want to give is for ammonium. And when we go through this process of developing generalized additive models for ammonium, again, outflow stands out as the single best predictor. But look at the shape of the relationship between ammonium and outflow, and I'm going to go back here compared to the relationship between silicate and outflow. Here we have a positive relationship because the rivers are a source, but for ammonium we have a negative relationship. And this reflects the fact that in this case, the rivers are diluting a point source of ammonium from the upstream sewage treatment plant. So we can get inverse relationships between outflow and nutrients or any other dissolved constituent, depending on whether the rivers are a source or whether they're diluting something delivered as a point source. So this dilution effect is the most important driver of change in annual ammonium concentrations. The second most important is total Kendall nitrogen loading from that sewage treatment plant. So we have a source term here, we have a dilution term here. They're both important, and this positive relationship between loading and ammonium concentrations in the estuary. Underlying that is a trend of increasing ammonium inputs from the wastewater treatment plant. So the ammonium trend of increase is a result of increasing inputs from the wastewater treatment plant over time. And again, I can hit the wrong button. Again, we can compare the effect sizes of these two processes, the process of river dilution versus a source. And in this case, the effects are about the same order of magnitude. So if we're interested in what's driving change in ammonium over this period of time, both things are important. The annual variability in outflow and its effect on dilution, and this incremental annual pattern of increasing nitrogen loading from that source. So the kind of here is that we can use pretty simple statistical models to both identify and rank the importance of processes of change that are captured in environmental time series. And I was a little bit surprised by the results here, in that between, 59% and 90% of the variance can be explained simply by two factors. So a lot of the variability in these water quality constituents that we've been measuring over decades can be explained by a couple of things. And notice that I've highlighted in yellow every single one of these constituents, from salinity to phosphate, has a component of variability associated with a hydrologic component. Mostly outflow, but for salinity it was inflow. And this just highlights the importance of variability in freshwater inflow as a driver of water quality variability in estuaries. And then I just repeat again something I said before, the relationship between, the sign of the relationship between ammonium and outflow and silicated outflow is different, and that again reflects the fact that that outflow is diluting ammonium and it's a source of silicate. So depending on whether a constituent is delivered by rivers or diluted by rivers, the slope of that relationship can vary in sign. Okay, now I want to do one last thing. So everything that I've talked about to this point is to try to understand variability from year to year over a 38-year period. So I did modeling analyses of the annual records, annual mean, annual summer chlorophyll, annual summer silicate. We can also use that same method of generalized additive models on the individual monthly time series. So now the question is, what's driving monthly scale variability of these different constituents? So I'm not going to walk you through the results of all of these analyses. I'm just going to show you the results from one to make a point, and that is the processes that drive monthly variability can be very different from the processes that drive annual variability, and we need to examine both. So the best example I can give is ammonium. So recall that when I did this analysis for ammonium variability, the two factors that stood out were outflow and loading from the wastewater treatment plant. But when we look at monthly scale variability of ammonium, temperature is by far and away the most important process. And so, and look at the relationship. So there's this strong, significant negative relationship between temperature, between ammonium and temperature. So what that's telling us is that during the cool season, ammonium concentrations are high. During the warm season, during the summer, ammonium concentrations are low. And one plausible explanation for this is the effect of temperature on nitrification. And so one interpretation of this graph is that ammonium concentrations are higher during the winter than they are during the summer because ammonium is being nitrified at a faster rate during the warm season than it is during the cold season. And that could explain this really funny seasonal pattern of the change rate of ammonium by month. This is the time series of nitrogen loading from the wastewater treatment plant. So we have this pretty fast ramp up of ammonium, of total nitrogen load. Actually, this is Kendall nitrogen loading over a period of decades. But even though we have this period of increasing nitrogen loading, for the summer months, remember I showed you that there's no trend of increasing ammonia during the summer months, but the trends are large and positive for the winter months. So one interpretation of this is that during the winter time, flows are high, nitrification rates are low, and ammonium remains in the form of ammonium by the time it reaches the sampling station downstream in the estuary. During the summer warm season, when flows are slower and residence time is longer, by the time ammonium discharged upstream reaches the sampling site, it's been nitrified to nitrate. And so again, key messages here: different processes appear depending on the scale of variability that you're looking at, and these kinds of modeling analysis can give us important clues about what's driving those changes over time. So this is kind of fast and furious, and these are the main messages that I want to leave you with. And I said this last night: estuaries are continually changing. River inflow is a master variable. Everything that we're looking at is related to variability of river flow, whether it's at the seasonal scale or the annual scale. We have pretty compelling evidence that water management has had an effect on salinity in this estuary. This relationship between this partial residual plot of autumn salinity versus the fraction of inflow that's exported. So, and it looks like the effect of water management is really only manifested during the autumn season. So we haven't had an effect, our actions have had an effect on salinity during the autumn season. That there are many different patterns of change. I talked about that early in the talk, and I'm just say this one last time, that outflow can either be a nutrient source or it can dilute a point source, in this case of ammonium nitrogen, and that we can identify that different processes of variability are operating at the annual scale compared to the monthly scale. So I hope this is giving you a sense of the amount of information that is contained in environmental time series and the kinds of analyses that we can do to incrementally extract further layers of information about what's driving what those patterns are and what's driving those changes over time. So thank you very much.
If you'd like to ask a question, I'll bring the microphone. Please use it.